Now on to the second of the video lectures associated with the nervous tissue, generation of action potentials. So we've already discussed how we get our resting membrane potential. That's due to the sodium-potassium pump and the leakage channels acting together to create the membrane more negative on the inside compared to the outside. We say that the resting membrane potential difference is negative 70 millivolts. Then we've looked at one type of change to the resting membrane potential difference, and that's a graded potential. And we saw where the graded potential is a localized change. It only occurs where the stimulus happens. We've seen how it is graded. A strong stimulus produces a strong graded potential. A weak stimulus produces a weak graded potential. And we've seen how it sums up. Now we're going to see the generation of an action potential. So action potentials initiate if the strength of the graded potentials by the time they diffuse through the cytoplasm of the neural cell body get to negative 55 at the trigger zone. So if you remember in our drawing of a neuron, we have all of our dendrites here in our neural cell body, and then we have our long axon over here, a few more dendrites, our long axon. So in our neural cell body and our dendrites, that's where we saw our graded potentials. So we had all graded potentials in there. They could sum up, so you might have a strong graded potential here, you might have a graded potential over here, and ions would move in, and then they're going to diffuse through the cell until they get to an area right around here at the axon hillock. That's where the axon joins to the neural cell body, and we call it the trigger zone. And if at that trigger zone we hit negative 55 millivolts, then you will get an action potential. All right, that's known as the threshold. So we can see, where is it, down here? The threshold for most neurons would be negative 55 millivolts. So if we hit negative 55 millivolts at the trigger zone, right here, then all the way down the rest of the neuron, down the axon, we will have our action potential. And our action potential is going to be due to the voltage-gated channels. All right, now an action potential, also called a nervous impulse. Nervous impulse is a sequence of events that will first depolarize and then actively repolarize the membrane to bring it back to more towards the resting state. And we're going to look at all the phases of an action potential. In an action potential, that's where we have our voltage-gated channels. So we have voltage-gated sodium and voltage-gated potassium channels opening in sequence. That means that first the sodium channels open up in response to hitting negative 55 millivolts, and then later on, slower, the potassium channels will open up in response to the same negative 55 millivolt threshold. All right. We call it, this has an all or none principle. If a stimulus reaches the threshold, you get an action potential. If it's greater than the threshold, you get an action potential, but it will always have the same amplitude. So a stronger stimulus will not cause a larger nervous impulse. All right, so here's our basic graph of the action potential, and we're going to come back to this graph and look at some key features. But first, let's notice down here we have the stimulus. All right, so we start off at our negative 70 millivolts. That's our resting membrane potential. Somewhere in either a dendrite here or here, wherever you want to pretend it is, or maybe right at the neural cell body, right? You get a graded potential, and it sums up until we get right here, where we will see our trigger zone. if we hit the threshold at that trigger zone, then we start into our action potential. So that's what we see down here with our stimulus. Right here, right there, it hits the threshold at the trigger zone. And if it does that, then we have our action potential. So we have three main phases of our action potential: our depolarizing phase, our repolarizing phase, and then something called the after hyperpolarizing phase, because it temporarily dips down a little bit below the negative 70 millivolts. Okay, now that's actually going to be pretty important because it's going to be related to something called the relative refractory period, but we'll come back to all of that later on. All right, so just to recall, the stimulus strength doesn't change the amplitude of the action potential. So remember, the amplitude in any of these waveforms is the difference between the base and the top. So that difference between there, that amplitude, that's the amplitude. The frequency is how many of those there are per second. For example, the ones in this blue range right here, you see that there's a lot more of those waveforms per time in millisecond. They're more frequent, so the frequency may go up. So if we have a subthreshold stimulus, that means it doesn't hit this threshold right here, doesn't hit the threshold, then you don't get an action potential. A threshold stimulus will take the membrane from negative 70 to negative 55. So if you wanted to do the math, negative 70 to negative 55 millivolts, that would be a plus 15 millivolt change. That's how much you normally need to hit the threshold. You need some stimulus that's going to change in a positive direction by 15 millivolts. Over here in our suprathreshold stimulus, you can see that we don't have any change in the amplitude. Remember, in the graded potentials, in the graded potentials for the amplitude, we saw that a greater stimulus caused a greater amplitude, but that was just in graded potentials. Here in our action potential, there isn't, that correlation doesn't exist, but what we do see is more frequent action potentials. All right, so let's just sum this up. I'm not going to spend a whole lot of time on this figure, but it's one that it would be a really good idea for you to go back to your book, because you can look through the reading and see what's going on at the same time. But what we're seeing here are our two voltage gated ion channels. We have our sodium channel, so we have our sodium channel, and our potassium channel. All right, so we have our two voltage gated channels right there. In the resting state in our sodium channel, so we were looking at our sodium channel, if this was our membrane, and here's my channel right here, and I continue on with my membrane, there are two different types of voltage gates. There's two different types of gates. There's right here, we have a, sorry guys, I'm having fits with this new writing device. We have what's called our activation gate. You can see it over here, activation gate. Our activation gate is closed, and our inactivation gate is open. Okay, so when you activate this voltage gated sodium, so this is the voltage gated, when you activate it, the activation gate will open up. So as we activate it, we see the channel down here. Once again, here's our membrane, here's our channel going through, channel. Now our activation gate is open, and also our inactivation gate is still open, so that's going to allow lots of sodium to enter into the cell. Okay. All right, then we've got to close the sodium channel and open up the potassium channel. This is going, so when we have the sodium coming in, we are rapidly rapidly depolarizing. So you see all up in here with that rapid depolarization. Now we begin to repolarize. So if we look at our sodium channel, here's our cell membrane, here's the channel, here's the cell membrane. So once again, this is the voltage gated sodium one. Now our activation gate is still open, but our inactivation gate has closed. So that means that sodium can't pass through anymore. But what is open now we have our potassium. So we have our potassium channel. So if we look at that potassium channel, it only has one gate, and now that gate is open. Right. Previously it would have been closed, and now it's open. We begin to see the initial bit right here of repolarization. Time continues, and you continue to see the repolarization continue. That means that a lot of potassium is leaving. So positive charges are leaving, and that repolarizes the membrane. Okay. Then at the end, that channel is going to close back. So the potassium channel will close. The sodium is going to reset itself back to the original phase, and we'll be ready to go again. Now I know that's a lot, but what I want you to do is to pull out your reading. There's a couple of nice videos of this on e-learning, also on Wiley Plus, and go through and watch what happens with all of these gates as we go through the action potential. All right, so moving back, first we had our depolarizing phase. So in our depolarizing phase, we hit the negative 55 millivolts, so a graded potential hit negative 55 millivolts at that trigger zone. That opens up our voltage gated sodium channels, and sodium rushes into the cell. The inactivation gate in the resting membrane is open, and the activation gate is closed. When we hit the threshold, both of those gates will be open and sodium will rush in. It's only open for a short period of time, and then the inactivation gate will close. So the inactivation gate will close just a few milliseconds later. Right? But a lot of sodium has rushed in, so you have a tremendous change in the voltage. In fact, it goes all the way up to plus 30. So we go first from negative 70 to negative 55. That opens up the voltage gated sodium channels, goes all the way up to plus 30, and our inactivation gates of our sodium channels will close. And this is an example of a positive feedback process, because if you depolarize one area of the membrane, the next area of the membrane will also depolarize. So this is how this voltage change will spread across the axon. Then next in our action potential, we had the repolarizing phase. So the voltage gated potassium channels also respond to a voltage of negative 55 millivolts. They're just slower. So that's why you get the sodium opening first, causing lots of depolarization. Then they close, and then the potassium channels open up, and a lot of potassium leaves the cell, and that causes repolarization. Okay, so the potassium outflow is going to return the membrane back to negative 70 millivolts, and in fact, it will hyperpolarize. Right, so enough potassium will usually leave the cell that could hyperpolarize all the way down to negative 90 millivolts before the potassium channels close. Now, once the potassium channels close, even if it's hyperpolarized, the normal processes for the resting membrane potential will return it back to negative 70 millivolts. Refractory period. Now, the refractory period is a short period of time where the neuron can't generate another action potential. We talked about refractory periods a little bit when we talked about muscle cells. So in a muscle cell, the refractory period was the period of time where you could not re-stimulate the muscle cell. Same idea here. period of time where you cannot re-stimulate the neuron, where it cannot get another action potential. Now, this refractory period takes two forms: the absolute refractory period and a relative refractory period. In the absolute refractory period, it doesn't matter how strong your stimulus is. This is absolute. You absolutely cannot get another action potential. And it all has to do with those sodium channels. I went into it in a little bit of detail. It's kind of strange that they have these two gates, an activation gate and an inactivation gate. But the problem is you have to return both of those gates to how they were originally in order to re-stimulate those sodium channels. So originally, if you remember here, we had our channel. This is our sodium channel. Okay, goes through the sodium, so it goes through the membrane. We originally, in the resting state, so in our resting state, if I can draw it in there, we have the activation gate. So I'm going to call that the ACT, A-C-T. That's going to stand for activation gate. So that's closed. And the inactivation gate, inactive, is open. So this is what you see in the resting for sodium. Now, by the time we get all the way through and we close this gate, so it activates and the activation gate is going to open up, so it'll open up, and the inactivation gate is open as well. Then when it closes, this is how the gates appears when the sodium channel first closes. The activation gate is open and the inactivation gate is closed. All right, so activation gate is open and inactivation gate is closed. You can't re-stimulate this. All right, the inactivation gate cannot respond to a change in voltage, so it just takes time. Come on now. It just takes time to reset back to the original. That's the absolute refractory period time. It's only when you get back to that resting position of those two channels that you can re-stimulate it. There's going to be a little difference between axons. Some axons that have very large diameters have a really short refractory period, so you can get a lot of nervous impulses per second. The smaller axons, smaller axon fibers, will have a longer absolute refractory period, and you get fewer impulses per second. Right. Then there's also what we call the relative refractory period. So in the relative refractory period, this is all about the after hyperpolarizing phase. So remember, this is the phase where you could drop maybe all the way down to negative 90 millivolts. Right? So normally, the resting membrane potential is negative 70. Right? We have to make it to negative 55 to stimulate the sodium channels. So that's from negative 70 to negative 55. That's a positive 15 change. But now if we're sitting at negative 90 millivolts, positive 15 is only going to get you, what, to negative 75 millivolts? That's not negative 55. You're not going to get an action potential. You would have to have a, what would it be, plus 35 millivolt change to get up to negative 55 millivolts. So it takes a suprathreshold stimulus, a really strong stimulus, to be able to start an action potential. So the potassium channels are still open, the sodium channels have closed, they've reset. You can re-stimulate it now. If those sodium channels have reset, you can re-stimulate it, but you're going to have to overcome this hyperpolarizing phase. Continuous versus saltatory conduction. Right. Now, so far, whenever I've described movement of this action potential down the axon, usually we describe it by continuous conduction. So in continuous conduction, we have our axon. So we have a nice long axon, right? And so we have some voltage gated channels here and they cause an action potential, and that will cause a change in voltage, and the area right next to them will detect that change in voltage, and they'll have an action potential, and the area right next to them will detect the change in voltage, and it will have an action potential, and it just goes all the way down, step by step, all the way down the axon. That's continuous conduction. Now the term axolemma, if you remember back from some of our terminology, axo means axon, lemma means membrane. So that's just talking about the membrane of the axon. Saltatory conduction is when the depolarization only happens at the nodes of Ranvier. Okay, so if we were looking at our axon here, and let's say that we had Schwann cells covering on a regular basis. So the spaces in between the Schwann cells are known as the nodes of Ranvier, and that's the only place where you're going to see voltage-gated ion channels. Right? Then this rush of ions in at the nodes of Ranvier will create a little current, and that will sweep the ions down to the next node of Ranvier, where it will depolarize the next node of Ranvier. Okay. All right, so these little boxes there are indicating the Schwann cells. So here's the example of our two different forms of the action potential. The first one, A, is continuous conduction. So you can see that step by step, every single portion of that axon experiences an action potential. But over on the second side, in saltatory conduction, you only have sodium coming in at the nodes of Ranvier. And then that will create a little current that will sweep the ions through the axon to the next node of Ranvier. So the next area will be stimulated, it'll detect a change in voltage, and it will have an action potential. All right? So this would be analogous to leaping from one area to the next. So the term saltation literally means to leap. That's going to speed up how fast the action potential goes all the way down the axon. So there are a couple of factors that will affect how fast the action potential will spread down the axon. So remember, the term propagation just means to spread. Okay. The amount of myelin. So if you have myelin, it's going to be fast. If you don't have myelin, it will be slow, right? Because it's going to depend on continuous conduction. Axon diameters, large diameter will be fast. Thin diameter will be slow. And then to a small degree, temperature. But this is not very important in humans. You may be wondering why I'm mentioning it. Well, what if a person had hypothermia? Hypothermia. Do you remember what hypothermia would probably mean? Hypo means low, thermia is temperature, so it's a low body temperature. Someone who's hypothermic often won't be able to think clearly, and that's because all of the nervous impulses, the action potentials in their brain have slowed down, so they can't think very well, and then when you try and control your muscles, you can't control them very well either because of the hypothermia. But normally, as long as you're maintaining a proper body temperature, it's not going to be an important consideration. All right, so we have all these different factors that can affect the speed of that nervous impulse going all the way down the axon. The propagation speed of a single nervous impulse isn't related to the stimulus strength. It's just related to those factors. If you're a larger diameter fiber, you're faster. If you're myelinated, you're faster. So we can break the different types of axon fibers out into 3 types. Type A fibers are the largest diameter. They are the fastest. They are myelinated. They're associated with the somatic nervous system. Type B and C fibers are associated with the autonomic nervous system. B fibers are myelinated, but they're thin. So they have one myelinated make them faster, but thin makes them slower. And type C fibers are both unmyelinated and very thin. So they are the slowest of all the fibers. All right, so I want to end here with this second video of the nervous tissue with a think, pair, share. Now I know we're not in the classroom, so I just want you to, if you want to pause the video and think about it, or write down the information and make sure you look up this information. So I want you to think for a second about what might happen if these myelin sheaths in either the central nervous system or the peripheral nervous system were damaged or destroyed in small areas. Think about it. What does the myelin sheath do for the speed of nervous transmission and making sure that the nervous transmission actually gets all the way down to the end of the axon? Right? So once you've thought about it, once you look it up, there's also a couple of diseases, and you can look these up in our textbook. Multiple sclerosis is associated with damage to the myelin sheath in the central nervous system. Guillain-Barré syndrome is associated with damage to the myelin sheath in the peripheral nervous system. And then our next video is going to begin with signal transmission at the synapses.